And without further ado, I'm going to turn now to our next speaker, who may not need much of an introduction in Queensland circles, but for those further afield, our next speaker is Emeritus Professor Frank Gannon. Now, I know Frank extremely well, having worked alongside Frank for quite a number of years. Frank is the former director and CEO of the QIMR Berghofer Medical Research Institute from 2011 to 2020. Before that, Frank was the director and of the Science Foundation Ireland, as well as having an illustrious career in Europe at leading up very large international molecular biology laboratories and organisations there. He's a high-profile scientist with a stellar publication record, but also a champion for science, and he's a fellow of the Academy. He's on the council of the Academy. He chairs the reports committee for our Academy. So Frank is a fellow who knows the literature. So his task this afternoon in 15 minutes is to help us get to grips with the literature. Frank, over to you.
Thanks, David, and hello everybody, and doing the usual check. There are different ways of approaching this particular topic. I did a bit of a scatter on it so that we cover different topics that are all around the question of getting to grips with the literature. I think just moving, yeah. So the first question is of information that we need to be aware of, and puzzling over that in the COVID context, I wrote an article, and I'm putting this down not as self-promotion, but to make some other points, which I called PRX3. And I wrote this article with PR standing for peer review, press release, and political response. And particularly if you're in the clinical area, and an area that's is of interest to everybody, including those who meet over a dinner table, there's a mixture of information and quality of information that you'll run into. The peer review is different than the press release, which is very often self-serving. And the political response, I won't say it's self-serving, but it is trying to integrate factors which are not all just the scientific aspect. And you see on the screen there that the paper is published in EMBO Reports. I should say, in addition to what David said, I've been an editor and founder of EMBO Reports, editor of Molecular Systems Biology and EMBO Journal. So I have been around all aspects, from papers going out to papers coming in on this. But you see that there are different links that you can get to get into the literature, and it's worthwhile knowing that they exist. And that will usually bring you to the paper of interest, unless you get blocked because of various reasons if it's not available to you or your institute. You'd also note that it was e-published in November 21 and then printed in 22. There was a time when I was a young scientist where the only source was print, and you read a journal from cover to cover, got all of the information in a diverse form. Now we can go in a more targeted way. We lose something along the line because we're not getting broad sources, but we gain speed and accuracy. Again, just to point out the types of paper, and there are primary data that are fully peer-reviewed, plus or minus editorially input and triaging. At EMBO Reports, EMBO Journal, an editor would get 3 papers on his desk every day. They would look at them at lunchtime. They'd come and say, guys, I think this is not interesting, because we've seen things like this before. It's not, and it would get rejected on the spot within a few days. So there can be that sort of triaging, and then it goes for the editorial or peer reviewing externally to experts. And again, that can be a variable quality, because you have to find person, the database is important, the willingness of people to review is important. And sometimes there's an editorial board, which in some cases is real, and in some cases is cosmetic. And say, look at all these wonderful people you've got behind us. Anyway, primary data, fully peer-reviewed, plus or minus editorial triaging is the top quality papers. Then you can get that to be single blind, where the reviewer is anonymous, double blind, where the author and reviewer are anonymous, open peer, where the comments are published alongside the article, transparent peer, where you anonymous reviewer report. So all of these things are moving towards added information and trigger some questions. If it's really a paper you're interested in, I'm not so sure about the conclusion was drawn there, and the referee may have asked a question, and you may say, well, I need to look at that further. The next area, which is growing of relevance, is the primary data in the preprint sites. Now, there, put up for initial surveying by the community, and then typically, and they should move into a peer review mode. And there are two that I mentioned there, BioRx and MedRx. What's important is that for ARC, famously, there was a big row about that this year, and now NHMRC are saying that they accept these as publications. And I've had one conversation where somebody was anxious to include a paper. It was not accepted in press, and I said, well, you can put it into BioRx, and at least it's accessible. At least the reviewers will be able to look at it, and that may be useful. Then, apart from the primary data, there are reviews which are sometimes invited and sometimes they're just submitted. There are editorials and opinions where people say what they think. They're more like journalism to a certain extent, and usually not peer-reviewed, although they can be rejected. I had one recently with a squabble over that PRX3, where the journal was concerned that I was sending a wrong message about the quality of peer review because I was pointing out it was slow compared to the needs. So there are aspects that will come on that as well. And then there are conference abstracts, which are usually not peer reviewed, very often preliminary, and very often further experimentation will do things. So you should be aware of what you're looking at when you're doing this. And the problem is that, as I said, finding a needle. There are 30,000 scientific journals. There are 2.5 million papers, one published every 20 minutes. In Pubmed, which is kind of the repository for all of the journals that we'd be interested in, there are 830,000 medical research papers published last year. And then there were a further 239,000 review articles. So you've got the information. The problem is you need to have the right magnet to find that needle. And the citations then add to it. But generally, 50% of the papers apparently are never read, according to one article that I read, and 90% are never cited. So it's not, when you put the literature out there, it is not necessarily going to have an impact because people may not read it. And you have to think about that, and I'll come to that point at the very end. It may not be read, and therefore what you've done is on your CV, but nowhere else. You haven't really advanced the area. As I said, Pubmed is a repository of all relevant literature. So I thought it'd be useful just to, again, to give you an idea of the scope. When I put in the search word cancer yesterday, that were the number of articles in 1 year. I came back about 2 hours later, and the number was higher. And so they're just churning in all the time. You put in cardiac, you get another number. Put in obesity. So one way that you may, if depending on where you are on your path towards a paper, you may need to say, well, what's known about the area I'm working about, and we'll hone in on that a little bit. So huge amount of information available there. And they're all then of the type that I showed on the first slide of the PRX3 article, where you get a DOI, and then that's an identifier of the information, and you click on that, and you get through to the paper or to the blockage to the paper, depending whether it's open access or not, and whether your institute has got a subscription or not. When you're drilling down, I thought this was an interesting example, because when COVID came out, first of all, I was in Ireland at the time, but you can be anywhere. And I was asked to get engaged about COVID and the information and sorting out all of the rumors. Remember Trump, the rumors from the realities, and is remdesivir good? Or it was a time of not concentrated data. But in the last year, when I went in again yesterday, there were 126,000 articles if you put in COVID. If you refine that and said COVID vaccine, and again, we keep waiting for information on the vaccine. There were 22,000 papers published on COVID vaccine last year. Refine it again and say COVID vaccine clinical trials, 1,500 almost papers in that. And then if you say, well, I want to know about the immune response, and you see clinical trial immune response, 429. Now you're getting into a more manageable pot of information. And then, because I'd read an article which I thought was interesting about the role of IgA in protecting individuals from COVID, I put in COVID vaccine, clinical trial, immune response, IgA. And there were 2 articles. And one of them was one that had been referred to in a press release that I'd read elsewhere. Very satisfying. So that way you can really hone in by putting in the right sieves of information. And I think that was a good and reassuring example, because then you can go back through the articles and do a different combination of search on the PubMed site and get to where the information you really needed. This may be a different way of approaching how do you get into the literature. So I took as a start point an area that my lab and that of Jason Lee was working on, G9A inhibitors. And so I just put in G9A inhibitor, and abracadabra, up came the paper that we published in the Clinical Cancer Research. Now I'm showing this again, not self-promotion, but you can click on that on the blue part there, and it will bring you to the paper. You can see not only the authors, and sometimes that's important. You may need to know what is Jason Lee working on at the moment. You may need to put his name in as a search. You can see the journal it's in, Clinical Cancer Research, and you can see the date, it was May 2021, and you got all of these connections. And you also get an entry into the paper with the abstract. When you've done that, you also get from PubMed further guides into the literature. So below the stuff that was at the top, there was a guide into similar articles, and you can see that there were 5 I showed on this screen, but it went further and further. And when you go into these articles, you'll see that there are directly relevant to what you were looking for. And then you start to have a choice to make, because you're getting a proliferation of opportunities to go after. Just one thing, the guide to relevant articles may not always be correct, because when you go into the PRX3 article, you end up also getting PRX3, which happens to be a homeobox factor, and it's not going to be what you're relevant. But these are relevant. And this is where you start asking yourself, well, which of these should I start looking at? Because a paper that's a relatively recent one, an older one, will have quite a lot of relevant articles. And it comes then down to the relatively tricky topic of impact factors of journals. Not all journals are equally demanding on the information that goes in there. That is not to say that every paper in a good journal is a good paper, or that there are not good papers in moderate journals. But it is a guide, and I'm perhaps because I was brought up on that, and I was also brought up within EMBO in the journals I was involved in wanting to know how good we were compared to the rest. So generally, any journal with an impact factor more than 10 is viewed as being excellent. So the paper that we published in Clinical Cancer Research is in a 12.5. So that was good. That's important to note, because when you go in looking for grants, the reviewers, although they won't say it, will probably be more impressed by an article in a very good journal, just because it's shorthand for this is a quality requiring article. If it's more than 3, it's good, and if it's around about 1, it's not so good. So if you look at those ones there, and I just did it a few seconds ago, you can see that the first one has an impact factor of 3.73, the next one 1.5, the next one 2.9, next one 3, the last one 7.65, and you can see that you'd look inevitably at the content, the title. Does this sound like what I'm really interested in, the aspect I'm looking at for? Or is it something that I just need to get as general information? And there's further information when you go into this, which may be a guide as to where you are going to put your paper. You might want to, people tend to, honestly, although they say ignore the impact factor, they tend to go for the highest impact factor, the most impact in their field. But they'll also then be looking at how long does it take for a paper to be accepted. And for the one which is +1, which is a popular one, which is the 4th one down there, on their website, they're very helpfully tell you that the average time is 171 days between submission and acceptance. 171 days, okay, so you're talking about 6-7 months. So you have to factor that in, again, if you're trying to get a paper out in time for a submission for an application. They also tell something interesting, which is desk rejection. You remember I mentioned about triaging? Well, the triaging is where there are editors who spend their whole time doing this. And although it's popular for the active scientist to say that those doing that work must be failed scientists. That is totally untrue. They are incredibly knowledgeable because they're doing it every day on broad areas and specific areas depending on how it's structured. But in +1, there's 25% of the papers are rejected at the desk level. They don't get beyond that stage because the editor saw no value in going forward with it. +1 accept about 50% of the papers that are submitted. And again, that's an important factor. A journal like Nature would be rejecting at desk level 80% of the papers. And what's important then is you get a quick rejection if you're going to go for some high impact journal. So these begin to play on how you're going to get there, but they also show something else, which I'll get to in a second, because first of all, continuing with the line on the G9A inhibitor story, they then in Pubmed give you the citations. Now this is, in fact, it's already out of date. Actually, it's now cited by 6, and that was in the last few days because it is something which is a recent paper. And you can see that there are 3 papers, and that's very important. So if you look at the third one, if you can see it, it says G9A, an emerging epigenetic target for melanoma therapy. That sounds interesting. So you'd want to know, what is that person writing in there? Is it a review, or do they have other information that you may need to know about? So you're able to, the G9A and cancer stemness and the promise of a therapeutic target. These are relevant papers that will guide maybe your next work, or may even challenge some of the work that you put out. It says, you know, they found that, but we didn't find this in a different cell line, therefore blah, blah. So that's the dynamic of research. And again, it takes you into it. And if a paper were older, it would have many more citations. And you can see all of the information nicely there for Pubmed. I think the real question I was asked, though, is how to get best value out of a paper. So this is my last slide, because this is an individual approach. People will go about this in different ways. And this is how I do it. I look at the title. If the title is something that tells me that sounds like an interesting paper, I should read it, I will go further. If it is a title which is intensely technical specificity, it may be the wrong thing for me unless it is exactly on the target. Then I'll read the abstract. What's this paper about? And when I was in EMBO Reports as the editor there, that was what I read. If this is true, is it interesting, was my question. And if it was interesting, that would have been the abstract. So again, getting into the abstract things which you want to effectively sell is important. I then skip to the discussion because I want to see where they ended up with in their analysis. And then I go back to the introduction. That always broadens your knowledge of the topic, and it also puts the research in a context. Then I go into the results if I'm still need to know that sort of degree of detail. And finally, I go into the methods if that's really important. So depending on what your purpose is, if it's a scatter that you want to narrow, you do it one way. If you already know specifically what you want, you'll be into the methods and the results faster than I've shown there. So that's an overview on how one can address the literature, and I'm over and out.
Thank you. That is a fantastic overview from a career in science. So thank you for sharing your tips with us. You covered an awful lot there, I have to say, which is good, great. But you covered the importance of, the quality of journals, impact factors, search strategies, how to sell your paper. So not just about reading the literature, but actually being part of the literature. And I think, that's really an important skill these days. One question I would like you to explore a bit more is, as a mere mortal human being with so much volume of material coming out each day, what's the best strategy, do you think, for keeping abreast of the literature day to day or week to week? How do you keep on top of your field?
This is where I think teamwork is important. I think when you're in your lab, you have a focus topic of the type that Lauren said, you're now working on this and you want to get into it. I think if you've got a team, they should all be reading aggressively every day, widely, and bringing new ideas in. And I think it's a good habit if you can set it up that in your lab meetings, which are an essential part of running your lab, where you interact and exchange initial information, that there is a component in there I read this interesting paper recently, or have you seen this paper? That's one part. The other part, of course, are meetings. If you go to a meeting, you get a array of things. But I honestly wouldn't say that I would sit down every morning and read. You wouldn't know how to start. So favorite journals. Over the years, I would always read Nature, I'd always read Science, I'd always read Cell, the table of contents, just to see what's in there, because I was brought up, I think, to be interested in different things and see where
days, Frank, we would go to the library and you'd look at the new weekly publications and you'd browse them all. Do you set up a or have you set up a an automated browser that you know lots of people have automatically, so that each morning in your inbox there's at least 4 or 5 new papers in your field? This is a new way of doing things.
Like a great idea, David. No, I didn't, and at this stage I won't. But it is a very good idea. Again, the problem is the search has to be accurate. It has to take what's needed in there. Just going back to the IgA, the example I gave about the IgA paper when I was tracing the COVID, one of the things that came out in that paper is that the data about IgA levels were sprinkled around other papers, but never highlighted. So if you were putting in IgA in COVID, you wouldn't get anything because nobody had highlighted it earlier on. Whereas if you're interested in IgA, you go into the papers and find. So it depends. There is a danger that you could spend all of your time reading and become
doing. Now there's a few questions coming in. So a very topical one. A concern with open access is the ability to pay for publication. What is your view on the impact of open access publishing on early career researchers where funding may be limited?
I'm nearly glad that you asked that. You remember I told you I was going to I write opinion pieces. So I've one that's festering at the moment, which is entitled "Apple Pie, Motherhood, and Open Access," because you cannot say a word against it because the language is so. Why not? I find it's a disadvantage if I don't have access to all the papers, but it has left out completely the component of paying for the right to publish yourself. So somebody has to pay for the process. There are people engaged all the way through. Somebody has to pay for that. And it used to be paid by the libraries, and then it became shifted a little bit to page charges, and then it got shifted to page charges plus color charges plus X figures free, and very complex, shifting the burden from the organization, the institute, onto the individual. And that has never been compensated. And it is a real cost. Annually, if you get a paper, even the ones that I published, they will say, do you want to pay €4,000, for a one-page article such as it's free? And I say no, because my institute wouldn't pay for it. It's as simple as that. Which institute? Anyway, so I think that it has been overlooked in the motherhood and apple pie reasons why open access is a great idea, but it is something that has not been structured properly.
Sounds like we could have a whole seminar on that very topic, a whole debate. So we'll come back to that another time. Last question, just a quick one, a 5-second answer if you can. Any tips on getting published when you're starting out?
First of all, don't believe that you have to know the editor. Just go on your quality, write it clearly. Remember, it's coming into people, referees. If it's not clear, it's going to be hard for them to get into it, and make sure that they get grasped by the abstract and the title.
Great advice. One other tip I was given was to pass it around to some colleagues who aren't co-authors and ask them to have a read of it and almost pre-review it before it goes in, have many critical eyes look at it. Yeah.